And you're certainly welcome to say hello to Bill, who is in McMurdo, waiting to present about what he's been doing. So good morning, good afternoon, good evening, wherever you are joining us from. And like I said, we'll get started in a moment. Give people time to settle in. Bill's class can say hello. You can all unmute and say good morning if you want to, or afternoon.
Hi, Sheila.
They miss you, Henske.
Oh, thanks.
Fun to see your
Mr. Dickinson's
students.
class. It is.
Hey, Bill.
Hey, Kevin, how's it going?
Oh, better than we
Miss you down there.
deserve. You're looking pretty good, man.
Thanks, you too.
You're looking better than the last time I saw you.
It's you know how the food is.
Oh, yeah, man.
I'm
Hopefully you're
being
loading
well
on the
taken care
pizza.
of.
We're excited to hear from y'all. Why?
Very good.
So quickly, Bill, why are you not up on the mountain?
Oh, that's a great question. Last time I was up there, the cord fell out and my hands are freezing, and I couldn't see the screen because the sun was either directly in my eyes or directly on the screen. So
Right.
I'm gonna try this way this time, and then I'm gonna give the mountain another shot next time I zoom.
Understandable.
The mountain's a lot of fun, but yeah, they want to drop you off up there with the shuttle now instead of driving up or hiking up, and so then they leave you there, and if things don't go right, you're just sitting up on the mountain with all your stuff for like 2 hours. So which is a fine adventure, but not when we have so many samples to analyze before we're done here. So hi everybody, I'm Bill Henske, and I'm the PolarConnect teacher that is working with the Dry Valley's ecosystem study this year. And you can see on my screen there's a picture of me and one of the members of our lab, Yessa Yorna, and down below there is a photograph of us in Beacon Valley, which is one of the most remote places that we visited while we're down here. I'm going to be talking about the process of coming down here, the preparation, the things we do down here, the things we found out, and then take any questions or fill in any details that you've been wondering about, if you've been wondering about anything. So Judy or Janet, if you want to go to the next. So I think they kind of already went over. If you want to ask a question, you can put it in the chat. I've got it open right next to my screen here. So just put your name, school, or institution. They're making a recording of this. So if you want to go back through and look at something later, or if you want to get a copy of the presentation, there's going to be a PDF posted as well. All right, Judy or Janet, you want to talk about PolarConnect real quick?
Yeah, sure. I'll talk about it. Good day, everyone. This is Janet Warburton. I work with Arctic Research Consortium of the United States, along with Judy Fonstock. Together, we administer the PolarConnect program. It's an NSF-funded program where we place teachers like Bill in the polar regions, both the Arctic and the Antarctic, so they can work with scientists and bring that back to you in the classroom. We're really excited to have an Antarctica program this year with COVID. As Bill will probably mention here, he was delayed for several years in getting to the field, but he's there now, and we're really excited to hear more about it. And with that, I think I'll just turn it back over to you, Bill. And one thing I just want to mention to everybody is, if you have questions as they come up, please use the chat feature for now. And if you want to ask them live at the end, you can raise your hand and do that as well. But yeah, we'll relay questions as they come along.
Cool. Thanks, Janet. All right, well, so here's me. So as Janet mentioned, I've been a Polar Trek teacher since 2019. Usually you're not a Polar Trek teacher for that long, but after our cohort started, that was right when we got hit with the pandemic, and we're still kind of in it actually right here. We're in a mask mandate down in McMurdo Station. Every time a new group comes in, you have the new opportunity to spread the little buggers around. So we just we have big vessel ships coming in right now, and with those big vessel ships we have big crews of people coming in. So in an abundance of caution, we've actually gone back to masking. So in this picture here, it's kind of talking about getting down to McMurdo and getting down to work in Antarctica. The last day before I left for Antarctica, it was -8 degrees in St. Louis. That's me in the frosty mustache and frosted glasses. I was trying to batten down my chickens, and the irony of coming here and it actually being quite a bit warmer was interesting. Then one of the interesting things for me getting down here was that I did not actually have a Christmas Eve day, so it just kind of went straight from the 23rd to the 25th when I was here. But I did actually get to meet Santa Claus. There's a Santa that's in the airport late at night, apparently during the Christmas season. And so at like 12 o'clock at night, I went and asked Santa some questions about which way he starts around the planet as he delivers presents, and what happens when he gets to that date line. And he just kind of like magicked off the question that I had for him, and wished me well. So all right, so it was over a full solid day just to get down to New Zealand, and from there we wind up in Christchurch, New Zealand, I think is our next slide here, where we get ready. So the U.S. Antarctic Program Center is actually in Christchurch, New Zealand. And when you get there, you do a whole bunch of training, and you do rapid COVID testing, and you walk around Christchurch trying not to get COVID, and you learn about the history of New Zealand and how it relates to Antarctic polar research. And they actually have a really big presence here in Antarctica. There's a base right next to McMurdo called the Scott Base, and that's the Kiwi Antarctic Program. In the picture there on the wall, you can see what's called the extreme cold weather gear. And when you go down to Christchurch, one of the stops that you make is the gear checkout, where you're going to get what's called Big Red. Big Red is this Canada Goose style jacket that is probably the warmest thing you can imagine wearing. Putting it on in mild weather, you just begin dripping with sweat. Nothing penetrates this jacket. It's pretty amazing. For any flights into or out of Antarctica or on continent, you're actually required to have all of your extreme cold weather gear, because the weather can just change at any minute. And it has before in the past, and people have been caught without the right equipment and gear. So that's a really huge emphasis. One of the things you'll see on the far right are the big white boots. Those are called bunny boots. And one of the interesting things about them is they actually use air as their insulator. It's like wearing 2 pairs of boots at the same time. We've probably all had a chance to like wear 2 pairs of socks. In this case, it's 2 pairs of boots with some air in the middle. And there's a little valve. And when you get on an airplane, you got to like open the valve so that they don't actually explode from the increase in or decrease in air pressure when you're up in the plane. So that's pretty cool. You can see the transport plane down in the lower right-hand corner. That is the New York Air National Guard, and their duty is in Antarctica right now. And they're the ones in charge of getting people on and off ice from New Zealand, and making delivery flights, and managing all of that air traffic. So that's a pretty cool operation for them. They had a neat little patch. I don't know if you guys have seen on service members' uniforms now, they have like a Velcro thing so they can put temporary patches. And theirs was of a Grinch, and it said Christmas canceled Antarctica expedition. So they were kind of getting in the spirit too for their actions down here over the winter time. All right, next slide. Yeah, oh, there's that patch. Pretty cool. So getting on the plane, you can see what I was talking about with the air pressure. They do serve lunch on the plane. The plane is a cargo plane, so you're just kind of sitting on either side in a big long line of people in all their extreme cold weather gear. I pulled out my muffin, and you can see what's happened is the air pressure from sea level. Once you got it up into the plane, my muffin package kind of exploded. So that's what, what that final leg looks like as we are going from New Zealand then to McMurdo Station. It's about a 5-hour flight across the ocean. Go to the next slide. And there we go. So once we get to McMurdo, it was actually unseasonably cold. So when we got out, everyone was really happy that we had been wearing our extreme cold weather gear, and unseasonably cold. So it was actually snowy, which is kind of rare here in McMurdo. It's across the McMurdo Sound is what we'll be talking about later, is the McMurdo Dry Valley, which is one of the driest places on Earth. Kind of extends to McMurdo as well, although we've had a couple different snow events. Even despite the really long travel days, one of the first things we did is take a big long tour around the town of McMurdo, which takes about 30 minutes to see just about everything. We're met by, when you get off the plane, the airport is really just a patch of ice out on the McMurdo Sound. It's actually the Ross Ice Shelf. The plane just lands out there, and you're met by this big monstrosity vehicle called Ivan the Terrabus, which is this gigantic tired transportation bus that picks everybody up from the airport or the strip of ice where the plane lands and drives everyone into McMurdo. Down in the basement of Prairie Labs, that's what that picture is there. It was one of the first things on our tour. You can see we were in a mask mandate back then as well. So despite the fact of getting away from it all and coming to one of the most remote places on Earth, we're still making sure to protect each other from transmitting diseases. And that's a touch tank. We had a touch tank in the lab down in Prairie, and they have creatures that the divers who go out underneath the ice will bring back for examination and study. So that's what I'm showing Abigail right there, is a gigantic isopod animal. And then down, at the bottom, you can see, I think that is, yep, that's me right there in the middle with my mask on. And that's Dr. Byron Adams, who is the principal investigator for our project, and that is his grad student, Abigail. And you'll see a bunch more pictures of them later as well. So I think there's a picture about driving out. Okay. So here's kind of like the people who make up our soils team. Dr. Byron Adams, he's from Brigham Young University, and he's formerly a marine biologist, and he's been working down here in Antarctica for about 19 years. I think this is his 19th year, and he's a pretty big celebrity here. Everybody knows him, and there's a reason, because he's a really amazing guy. His co-conspirator is Jeb Barrett, who teaches out at Virginia Tech and also kind of in the same fields. Yeso Yorna is a student of Dr. Byron Adams. He's a PhD student, and he is actually running the labs this year. So he came down really early, set everything up. And so all the experiments and all the procedures, protocols, helicopter requests, packing orders, even down to what food do you want to eat out in the field, he's pretty much in charge of. Next, I mentioned earlier, was Abigail. She is doing a really cool study where she's trying to look at the genetic variation of one species of nematode and how it differs from different places in the dry valley. So we'll talk about in a little bit the relationship between creatures in the dry valley and the rest of the world. But if you are a microscopic organism and you're in one valley, there's virtually no way for you to interact genetically with the creatures in another valley, because there's huge glaciers and mountain ranges in between, and there's very little time in a season for you to migrate as a microscopic nematode from one place to another, and there's no bird mechanisms or anything else. So she's looking at the genetic differences between the valleys. She can tell how many years they've been separated genetically. Next, we have Sarah and Meredith. They're Dr. Barrett's PhD students in his lab, and they're looking at ground truthing the biological communities using satellite information, which is really cool. If you go to my journal, one of the last couple ones, I had an entry on the work that they're doing, looking for biocrusts. And then finally, we have Ariel Waldman. Ariel Waldman is an artist who came down a few years ago as an artist in residence, and she's doing a documentary this year, which is really cool. She's looking at the scope of life from the most minute to the most gigantic. So from a submicroscopic level to the macro level of planets. So she's got some really cool stuff, and there's a link at the end to some of the work that she's been doing. So she'll be putting together a lot of the work that we've been doing here as a little documentary series. And then, of course, myself, Bill Henske, I'm a PolarConnect teacher. Next slide. So here's what we're doing as a lab, or as a group of scientists with the project. Our main idea is to use the soils of the Antarctic dry valleys to figure out how ecosystems actually work. The unique thing about the soils in the dry valley is their biological communities are really simple. There's bacteria, there's cyanobacteria, there are 4 or 5 different species of microorganisms beyond that, and that is about it. So you can pretty easily figure out most of the constituents of the biological community, and then from there you can track the way they exchange matter, such as carbon, nitrogen, phosphorus, etc. The soils team is part of what's called an LTER. And the LTERs are these sites that the National Science Foundation has funded around the United States. And they're places where we're gathering information about how the environment is changing or acting over time. So in some cases, the environment doesn't change over time. But in our particular case, we're looking for how ecosystems respond to changes in the environment. So like increased temperature, or decreased temperature, or increased moisture, or decreased moisture, increased wind, decreased wind. But in other LTER sites, they might be looking at changes in nutrients in the soil or changes in gases in the atmosphere. You can see in the bottom picture there, that's a picture of what dry valley soils look like. You can kind of see a little bit of a glacier in the background, and there's these little cones that look kind of like the cones you put around a dog, so a dog doesn't like chew on its leg when it's not supposed to. But anyway, those are soil heating cones, so it allows us to, like, in very small increments, change the temperature of the soil so that we can over time collect samples from there and identify and describe the organisms that are there versus a place that did not have the increase in temperature. So that's one example of a temperature gradient, where we're changing the temperature of the soil and looking at how it affects the biological community. Okay, we can go to the next one. So here's a picture of the dry valleys, and just for reference on the on the right-hand side of the map, about 30 miles away is McMurdo, and in between is the McMurdo Sound. And I'll show you, I've got, if you can pin my camera here. Janet, are you able to do that? I got the window here.
Yeah, you're pinned
Okay.
already.
Okay, can you see the sound out here or no?
Yes, we
So we
can.
can-
And we
okay,
see the
okay.
mountains too. It's
Okay,
beautiful.
cool. Yeah, fortunately, it's a really nice, clear day. These are the Trans-Antarctic Mountains. And that's what makes the dry valleys the dry valleys. As you know, most of Antarctica is covered by 1 to 2 mile sheet thick ice sheet. And it's a glacier, so it's moving. So as materials are added to the glacier, that ice sheet moves and it's moving downhill towards the ocean. Well, here around McMurdo, there's the Trans-Antarctic Mountains, and that prevents that ice sheet from just coming through towards the ocean. So here you can see that these mountains, they're not all solid, covered in ice and snow. There is ice and snow, but they're not covered in an ice sheet like the rest of Antarctica. And that's one of the things that makes this area unique. Oh, you can also see, oh, the icebreaker took off. One thing while we're here, you can see there's open water right here. It actually wouldn't be open right now, except for the Coast Guard just came in and has opened up that area so they can bring in next year's food supply for the entire station. So this is the vessel time of the year when big vessels come in and they unload supplies for next year, and they take back things like scientific data and samples and equipment that needs to be repaired and stuff like that. So anyway, so that's the where we're working is the McMurdo Dry Valleys, and specifically we've done most of our work in what's called Taylor Valley, which is kind of right smack there in the middle. Let's see. Okay. Next slide. And you might have to go forward one too. I think it might have had a, yeah, that's what's going on. Yeah, keep going all the way. Okay, so the first Antarctic explorers in their notes, they all pretty much said the same thing. There's no life here in Antarctica. Now, of course, everyone knows about penguins and seals, and surprisingly, when we look out today at the Sound, there weren't any seals, but they've been kind of freaked out lately about all the ships coming in, so they've gone to the other side of Ross Island. But they really weren't looking for the kind of life that can exist here in Antarctica. And the more we've looked, the more places we've found examples of life. And here's a few that we'll be talking about. The first one is probably the largest by biomass source of life in the dry valleys, and that's the microbial mats on the bottoms of lakes. These microbial mats can be really thick, and they're protected from the extremes of the environment by 3 to 4 meter thick layers of ice, and they're constantly kept moist because they're at the bottom of a lake, and they have plenty of light because the ice is transparent. So these have been able to form over thousands and thousands of years. They grow really slowly, but they're the basis of a whole food web in the Antarctic dry valleys. The next place where we find life in the dry valleys are the streams. The streams are not rivers and streams like we think about back in the United States. These are very temporary. They're only the result of meltwater, which only happens at certain times under certain conditions, but it's enough to wet the soil, and liquid water is what living things need to exist. And so streams are an important area to find life that's thriving in Antarctica. Another really interesting place is glacier ice. So glaciers, as they're traveling through the dry valley, well, even coming off of the polar plateau, they're accumulating sediments and windblown debris. And when that sediment or wind-blown debris has sunshine shining on it, because it has a darker color, it absorbs more of that solar energy. It then actually melts its way into the ice. And because it has a darker albedo, it will melt some of that water and it'll create a little pool. And in that little pool, it can be an incubator for living things. So those are really interesting places to find life forms. I think we're missing one more step forward, Janet. I'm not sure. Yes, in rocks. This one I thought is really, I'll go back one. Life forms actually living in the rocks. In some places in the dry valleys, there are these really porous sandstones called Beacon Valley sandstone. And the Beacon Valley sandstone has these organisms called endoliths that live inside the rock itself. Outside, you can't see them. But if you break it off, you'll see a little thin layer or band of cyanobacteria and other organisms that can chemosynthesize and photosynthesize. They're actually living in the rocks. Of course, you've probably seen lichens before on rocks. They're not plentiful, but they're out there, very slow growing. And then our focus, which is the creatures that live in the soil. You can see there's a picture of a nematode and a happy little tardigrade. So that's what our focus is actually on. Next. All right, so here's what we do, as you can imagine. So there is Sarah. She's going in our refrigerator there, and she's got what looks like and is hundreds of different samples of soil that we've collected. So in her particular study, what she did is got satellite imagery data and uses the reflectivity of the surface to identify what wavelengths of light are coming off. As if you were looking at a satellite of Missouri or Brazil or something, and you saw green reflecting off, that would be a pretty good indication that there's forest or grassland there. In the case of Antarctica, there aren't places where lots of green light is reflecting off. Plants and photosynthetic organisms down here don't have green pigment on the outside for sure, because that green pigment is not very useful with the high amount of ultraviolet radiation. They actually have to produce other pigments to block those wavelengths of light. So they're looking for different kinds of colors that are reflecting off. And there we got other things happening in the Prairie Library Lab here. So anyway, what they did is they identified 64 different spots where they could possibly find what are called biocrusts. Biocrusts are organic communities that are microscopic and just living in between the particles of soil. So we went to different places, and you can see the really high-tech way that we take our soil samples. We have a scoop and we have a sterile bag, and we scoop it into the sterile bag, packages up, put it in a cooler, because despite how cold it is, we actually have to prevent it from warming up in the plastic bag. Next. All right, so when we do go out in the field, actually there are camps that are set up by the National Science Foundation and managed by the Arctic programs. And this is an example of one of them. I think this is the Lake Horror Camp. And you can see there's little lab buildings where scientists can conduct their research while they're on site. So there might be one team staying out there that's studying the limnology, another team that's studying streams. And on the inside, there's a hut. And in this particular case, there's a camp manager, because it's kind of a busy camp. So they would have multiple different teams coming in at different times. And it was just easier to make sure, because when you're in a camp, you're in charge of taking care of it. So making sure that it is kept clean, making sure dishes get done, making sure that materials are organized, poop buckets are cleaned out or sent back on the helicopter, etc. Everyone staying in camp is staying in these 4-season tents, which are really cool. The Lake Horror Camp did have an indoor area where we could watch the movie Blood Glacier, which is not very realistic of a horror movie. But the dry valleys have a geological feature called Blood Falls, which is probably the original idea behind the movie Blood Glacier. So that's what life in the camps looks like. And if you want to go to the next one. Okay, so once we get all our samples back, we have to get all the critters out of those soil samples. So there's a picture of me literally mixing the soil sample with water and putting it through a sieve. The sieve has different size openings so that big stuff gets filtered out, little animals go through, and then they get caught on another sieve. And then we take those poor little guys and we put them in a centrifuge. We spin them around. All the animals go to the bottom of the centrifuge tube. All the water comes to the top. And then we take the centrifuge tube, we mix it with sugar water, which is denser than animals, put it in the centrifuge, and then all the critters are on top of it. And that's how we get our animals out of the soil samples. So kind of fun. Next step. And then we have to count them. Fortunately, there aren't that many organisms in a sample of deserts or a polar desert soil. However, depends where you are. In some places you might find in 100 gram soil sample, you might find 20 or 30 different creatures, but in some we found over 10,000 different organisms in just one little sample. So you can see in the lower left-hand corner, that's called a counting plate. We take that centrifuge tube that has about 15 milliliters of water and animals, we pour it in there, we put it in the dissect or the binocular microscope, and we use little clickers. So every time we see male nematode of Eudora lima's genus, we click that. Every time we see a juvenile, we click that. Every time we see a rotifer, we click, and tardigrade. And sometimes it's a click every 30 seconds. Sometimes it's click, 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 where the soil is really full of life. Usually it means that soil came from a really moist environment, like near the algal mats around a stream or something like that. All right, so we have data on their species, their sex, and then their age too. You can, yeah, next one. And then we have to put all the data in so that we can analyze it. This isn't that bad of a deal. This is actually what allows us then to analyze it. You can go to the next one. All right, so collecting all the data, we collect data from different sites that have different specific experimental designs. Like I said before, some of them are set up to look at a gradient of elevation, or might be set up for a gradient of moisture or temperature. But by collecting all the background data, we can then use the data to answer other questions as well. So like, how are the communities just changing over time? This is a big, broad, general question is, are they becoming more diverse? Are they becoming less diverse? Are they becoming more numerous? Are they becoming less numerous? How is the diversity changing over time? How does elevation or distance from water or temperature or the nutrients that are in the soil actually change the community itself, or does it change the community itself? You can change the next. Okay, so here's kind of like the basic food web here. And down at the bottom, it's kind of a fuzzy set. So we have the microbial community. The microbial community could be algae, it could be mosses, it could be cyanobacteria. Usually it's a combination of all those different things. And then from there, we have the nematodes and the rotifers and the tardigrades, things that are primary consumers. But in most cases, they're not primarily primary consumers. They are mixotropes. They may eat all over the food chain, depending on their stage of life or the food availability, et cetera. So despite the fact that it's a very simple food web, it's actually kind of complicated because in some cases they switch around their roles and do different things in the ecosystem depending on what's available. Famous quote there down at the bottom is that the nematodes are McMurdo's equivalent of elephants and lions. These are the biggest terrestrial organisms on the entire continent. If you look at my last journal post, actually, we did find the largest terrestrial organism, which is called a columbola or a springtail, and they're less than 2 millimeters. So even though when you see them, you can see them. You don't even need a microscope. You can just say, oh, there's one. But it's pretty hard to point out to someone else that you've seen it as well, because they are quite tiny. And those are the largest ones. All right, we can go to the next one. All right, so here's our overall observations over many years, and this is what we would expect to see. So as it gets colder and drier, there's one particular species of nematode, the Scotinema, and it's a generalist. It does really well. It's the predominant species of nematode throughout the dry valleys. And what we would expect to see with warmer or wetter conditions is an increase in almost all the other creatures. So the Plectus nematodes, the rotifers, the tardigrades, etc. So, and that is what we see experimentally when we do the alteration experiments with temperature and moisture. And you can click forward on that one. Oh, yeah. So here's kind of like a sketch of what an experiment looks like and the predictions that go along with it. So we expect to see, with changing climate and changing condition, a decline in the dominant species and an increase in the less common species. We expect actually diversity to increase with warmer or wetter conditions, and overall we expect abundance to go down though. So it's kind of like the other side of it. If the dominant species declines and the subordinate species increase, overall the total biomass will decrease. And so here experimentally is showing the same thing. So we're looking at the Scotinema declining actually over time, especially at higher elevations. So they don't seem to be declining in areas that are, and by elevation, it actually means distance from the lakes or the streams with available water. So go to the next one. Okay. Oh, yeah, go to the one with the colors. Yeah, that one. Okay. There's a really interesting paper you can see from 1990 to like 2004 or something like that. It actually was getting colder in Antarctica. And when that was reported, some people in the media were going a little bit crazy, like, oh my gosh, global cooling. And it is true, it's a long-term trend, but it actually was, there had been a trend of increasing lake levels, and increasing lake levels means that there's increasing availability of liquid water, which means that all of these changes that we are predicting would happen. And then there's this long period of fairly stable to declining temperatures over time, and then it picked back up again. So what we are looking at is a continuation of that trend as well. You'll notice that these are all the different lakes in Taylor Valley. They're not all increasing in height at the same rate. Some are increasing much slower. It depends on how much meltwater is coming off of the glacier that's feeding them, and then the porosity of the soil and other variables like how much can sublimate from the soils into the atmosphere. Oh, and another interesting thing about this, and then the next one, is trying to parse out what's actually causing that. And the most recent evidence is that it's actually being caused by solar maximums of ultraviolet radiation is what's leading to the rapid melting of the melting events that are happening on these glaciers and these glacial valleys. So you can see in this particular slide, it's showing that 10-ish year period where the temperature was slightly declining and stream flow decreased. That's because with lower temperatures, you have less melting. So with less melting, you have less stream flow, and with less stream flow, you have less water available. So that's one of the big trends with and with less water available, you have less biomass in the soil. That's less animals and plants that are actually living in between all those little particles of soil. All right. So I think it was good. 7:40. Time for questions. So these are links to the journal. I said that's a link to Ariel Waldman's site where you can check out the work that she's been doing on microscopy and macro and micro stuff with the Antarctic ecosystems. And then if you have questions too, how you could contact me if you'd like.
All right. Yeah, thank you, Bill. And I think Judy, you can stop. Yep, you did stop the screen sharing. So feel free to turn on your videos here if you want to. And also, if you have a question, there is a reaction. You can actually raise your hand, and that lets us know that you have a question that you want to ask live. Otherwise, just put them in the chat. There were a couple of questions that came up. How thick is the ice right off of the sound, you know, that the icebreaker is trying to break through?
It actually varies because the ice is not like one uniform layer, because it's been broken up and then it doesn't all melt, so then it refreezes together the next winter. So you actually have kind of patchy ice distribution. So some ice might be a foot thick, some might be 15 feet thick, kind of depending. In general, it's 2 to 3 feet thick, I think, where the icebreakers are coming through. The boat makes it look like it's just like ripping through a sheet of paper. The only time I've seen it is from helo flights overhead. So it's hard to get an idea of it. They don't really want you walking around out on the ice. But yeah, it's between 2 and 3 feet thick in a lot of places. Where they break it up, though, is kind of cool, because that provides air holes for all the marine mammals. And the first couple passes through, when the Coast Guard ship comes in, we saw fin whales and orcas, and that's a few penguins pop up. And prior to that, all you would see is along the sea ice cracks, you would see, I think someone asked what kind of seals. The seals we see most often are called Weddell seals, and they actually live on the sheet ice. And it's inexplicable if you don't know what to look for. They're just like, they look like slugs, like laying out on the ice in the middle of nowhere, but they actually have tiny little Weddell seal size holes that they come up through. And sometimes working in the lab, you'll look out the window like I showed you earlier, and you'll just see dozens of them all lined up along a crack, but you don't see the way in or out. And then you'll look a few minutes later and they're just all gone. And it's fun because at some point they all just must have decided to all dive in when you weren't paying attention. And so anyway, yeah, they're pretty cool to watch.
Yeah, that's cool. So let's see. From Lucy Coleman's class, they want to know which sample site was your favorite.
Ooh. My favorite actually was one of the worst. Oh, my favorite site was called Horth Hill, and you actually may be able to see it from here. It's right off of it's the Big Bear Hill right across the sound. That's actually about 20 miles away there. And the reason it was a favorite is probably for my own bias, is that we hiked in a little ways, and up on a hillside there was snow melt. And so I was kind of thinking, the snow melt meant colder and colder and colder, but what it really means is moisture conditions. And there was a tiny little meltwater stream, but it was just lined with bright green moss, which is not very common at all. Usually what will happen, if moss does get a little foothold somewhere, it'll get covered in sediment. And then the next year, the moss will grow up through the sediment, then it gets covered again. So you get these little mounds. And by little, I mean like little mounds of moss. But that's the only green you see. So it really stands out, and it's really amazing to see in the context of everything else being, dark black rocks and white ice. So I think that was one of the coolest sites. The other site that I thought is probably my favorite is Beacon Valley, which is a place where NASA has done a lot of testing for their remote vehicles because it's the most similar environment to Mars, apparently. That's a place where we did find the endoliths, which are the organisms that are growing inside the rocks. But Beacon Valley is by far the driest, coldest, windiest place that we visited. It was very extreme. But even there, we found organisms living in the soils, any place where there was meltwater that was actually able to. So the problem is water itself isn't good enough for life, because what tends to happen is that when the water flows, it then carries nutrients, and then the water evaporates and leaves all those nutrients behind. So anywhere where water is flowing and then collecting tends to become toxic to organisms, because there's so many salts and other chemicals then in the soil, and there's no plants that would normally use those up and convert them into useful things for other organisms. So here you have to have just the right conditions where there is liquid water, it is flowing, but it's not like collecting in one place. So there's a few of those places in Beacon Valley as well.
All right, she said, thanks for sharing. I haven't heard of Beacon Valley before, a Mars analog. Thanks, Lucy. We have a question from Robert. In the slide where you showed sample collection, that sure looked like liquid water. Is it?
Which one was that?
It said in a slide where you showed the sample collection.
Oh yeah. There's definitely liquid water. So the different lakes throughout the dry valleys they may be, some of them actually will melt out from the edge inwards, and that's what's called a moat. And so the ice they're never free of ice, but they'll have like a little patch that goes from the edge of the lake, maybe in some cases several feet or several yards even, to the ice itself in the middle of the lake. Some lakes never unfreeze at all. So yeah, there is liquid water. We actually visited a couple days ago the largest river in Antarctica, called Onyx River, and it's like 12 miles long, and it's only a river when it's the summertime here, and you could easily jump across the largest river in Antarctica. There was a big melt event a few years ago, by a few, I think like 10 years ago, and the Kiwis actually allegedly rafted the Onyx River when there was a large meltwater event. And I think you're not allowed to do that. I think the Antarctic Treaty probably like bans like recreational activity. Maybe they had a scientific purpose and had a research hypothesis or something like that. But that is the story, that they are the only people ever to raft in Antarctica. Sounds pretty cool.
That does sound very interesting. Melissa is wondering, are mosses the only plants, no vascular plants, right?
Yeah, there are no vascular plants, although there are some really good pictures from the limnology and microbial interactions team showing the algal mats underneath the lakes. And some of them kind of grow up from the bottom in like big wispy things that would remind you maybe of corals, in the way that they can even branch out and take up more surface area and have more photosynthetic capability. So they kind of like have some similarities, to what vascular plants do, although they're not vascular plants. So yeah, our biggest plants are mosses, and those
That's
mosses
interesting.
are 1 to 2 millimeters in height.
That is interesting. Kevin and his students we can tell from your smile and happy attitude that you are loving your work there. Has everyone stayed as happy and healthy as you? Also, what was the toughest thing so far? Did you get to use your diving skills down there? You are looking good, my friend.
Thanks, Mr. Dickerson. He was one of the first people I ever talked to about coming down here, and he's an alum as well to the Polar Trek program, and is very inspiring to me. And I really appreciate, he gave me a lot of advice and help before I came down. Sadly, I did not get to use any of my diving ability. When you meet the divers here, you realize what league they're in. When they are doing dives out on the lake or out in the sound, they're going through two like holes in the ice that are body diameter, but they're going through like 15 straight feet of ice tunnel to get down to a place in the lake, like just a few feet between the ice and the bottom of the lake itself. And the bottom of the lake is covered in sediments, and I just thinking about it gives me anxiety. They are an amazing crew though, and I've never met like more happy, well-balanced people either. They were out in one of the camps with us last week and just having a good old time and talking about everything they found, and who forgot to zip their dry suit up and who didn't, and who may have fallen out of the boat into the moat on the way out to the dive site. So there's a lot of opportunities to do some really interesting science, and that is one of the funnest ones, is the dive teams that are out looking at the microbial communities on the bottoms of the lakes. That's super neat. Earlier in the season, for everyone else, they have a dive tube that they put out, and they drill a big hole in the ice, and they put this tube down into the ice that you can actually go in so you can see underneath the ice. And there was some cool footage someone took of a adult male Weddell seal like going up and showing off for the people in the dive tube earlier this summer. So it was pretty cool.
That is awesome. Great descriptions of everything, and some comments about how lucky you are to have seen the Onyx River and get the grand tour. So is there any other questions out there, especially from people that have students in their room? I want to make sure we take care of that. I know there's a few family members and fans, Bill fans out there. So anything else? If you didn't get a question answered, or your students all departed, I know, I think Bill's class actually had to go to lunch. So, sorry that your students took off. Anyway, what's your plan now, Bill? Can you kind of give us a synopsis of what's happening, and when are you leaving?
Yeah, well, I'm not saying it's not fun to do all the lab work, but going out in the field and doing the collection and hiking around the dry valleys to the different sample sites and heloing into remote places and checking them out, trying to find new places, like that is really fun. But what that means is we now have hundreds and hundreds of samples to analyze before we leave. You can't just leave the creatures laying around in bags. That's not their favorite habitat. They would rather be in the freezing cold of Antarctica than in this plastic bag of soil in a refrigerator. So we have to process all of those. as quickly as possible. And after we process all of those, then we have to preserve all of those specimens. So it means that we're either going to freeze them down in little tubes so people can sample their DNA, or we're going to cryovial them, which means that, remember, these animals can survive like dehydration. They can dehydrate themselves and then freeze solid, at -40 degrees. So we actually will do that with our samples, and then when we bring them back to the United States, we can revive them and we can culture them, or we can observe their behavior, do a number of different things. But right now, a lot of the research that's going to happen later on is genetics based, looking at the relationship between the genes of the organisms and the different valleys and how they differentiated over time. There's also work that's going to be done on like a molecular clocking them, basically seeing how long they've been a divergent group of organisms based on the number of mutations that they have. But yeah, here we're just going to be extracting, counting, recording. We probably won't even have a lot of time to, I mean, you kind of analyze stuff as you go. You're like, that one had an awful lot of male nematodes. Where was that again? But the detailed analysis is probably all going to happen once we all get back to our different universities and schools. So.
Yeah, and hopefully
to
you'll
do.
get it. Yeah, hopefully you'll get a chance when you get back at some point to kind of see that process in action as well. So
Yeah,
the
for sure.
second stage.
Yeah.
And
Pulling
so
out a
when
lot
are
of
you
data.
leaving? Oh yeah.
Oh,
I was
so
when are you leaving?
yeah, pulling out a lot of data sets for student groups to use and analyze too. That was one of my side quests down here, was to talk to different scientists about what their main ideas were, and what kind of data that kids could look at to draw conclusions or test hypotheses, et cetera. So kind of working on that as well. That was someone, Melissa. Are you going to bring back some dehydrated tardigrades and rotifers? Yeah, we could actually probably work with Dr. Adams's lab to get samples world packed for student analysis as well. I'll have to talk to him about that and see what's good with it and what is not good with it. So it could be something we can do with our permits, or it might not be. So.
And Judy's asking, did you get your postcards sent, or did you do any of that?
I did send all my postcards, and I sent them like 3 weeks ago. So we'll see. Like they were actually out of postcard stamps too, so I had to splurge and get the full price. Set me way back. Wasn't like Elaine. So if anyone was following Elaine Krebs's South Pole adventure, she had her maybe her postcards were thicker or something, but no, she had like six times as many postcards as I did. So she was doing pretty good. Her hand was all cramped up from all the writing. So
all right. So yeah, we're gonna wrap this up. If you didn't get your Judy, we go back to the screen sharing real quick. If you didn't get your questions answered, feel free to post them to Bill's journals, and he's going to be headed back. I think he's staying there till February 6th or so he's still got some time there in Antarctica. As we started this presentation, we are doing a recording, so if you need to share it with other students or share it with somebody else, anybody that registered will get a copy, as well as it will be posted on our website. Judy is sharing also other ways to join PolarConnect. Again, Bill's here, will be doing journals for a little while. He also had some videos he wasn't able to share with us today, so you'll want to check those out on the YouTube channel or in his journals. And we do we might have a expedition this summer in the Arctic. So this is how you can stay connected with PolarConnect. And with that, I want to thank all of you for joining us today. And thanks to the National Science Foundation for funding PolarConnect, and to Arcus for hosting it. And to all of you for joining us, and good luck with the rest of your adventure there, Bill, and we look forward to reading more about it.
Awesome. Thanks everyone for coming. I appreciate it.
Yeah, and for those of you that want to hang out and
All right.
say hi here, feel free.